help alleviate that tension. And that's what topoisomerases do. And then you have these single-stranded binding proteins, the really long name here. What do these SSEBPs do, the single-stranded DNA binding proteins? They help hold the double helix apart because when the helicase is unzipping it, the attractive force between the nucleotides wants to bring the two strands back together, and the single-stranded binding proteins help prevent it from slapping back together. Then you have, next coming up is a primase. So what is this primase doing here? This primase is creating an RNA primer. So it's building the RNA primer because the DNA polymerase, which is going to come in next, has to have a free hydroxyl group to bind to. And so you have these RNA molecules being set on the DNA strand so that DNA polymerase is primed and can grab onto the DNA strand thanks to the RNA primer that is there. And so the primase is making the RNA primer so that DNA polymerase, which is the big green guy here, is going to be able to sit down on the DNA now, thanks to the primer, and start replicating the DNA. So DNA polymerase 3, that's the really important guy here, that is putting in the correct DNA nucleotides. And it's doing this on the lagging strand and the leading strand. Leading strand template. So remember we had two strands we're working off of, the lagging strand and the leading strand. The lagging strand, it's continuous in the 5' to 3' direction. The lagging strand is also doing it in the 5' to 3' direction. But because of the way the helix is being unwound, it has to do it in what I call a piecemeal kind of way, making Okazaki fragments. So the lagging strand makes these Okazaki fragments that have to be annealed. Annealed is just a word that means brought back together. When you anneal something, you bring them back together, and that is what is going to develop the ligase, which I don't see on here, but there's going to be a ligase that's going to come in and help cement or bring these Okazaki fragments and glue them back together once they're created via the lagging strand. Then what's not on here is DNA polymerase 2. Sorry, not DNA polymerase 2. DNA polymerase 1 is going to remove the RNA primers. And again, when it removes them, the gaps will be filled by DNA. And that's going to be DNA polymerase 1 is going to remove the primer and put in the correct nucleotides. And then this last thing is DNA ligase is going to come in and help seal everything. It's going to seal all the gaps between the Okazaki fragments. Any gaps that might be there is going to be sealed by the DNA ligase. DNA ligase kind of glues everything together and makes sure it's in, there are no gaps, essentially, between any of the strands. So, all right, with that review, I promised you we were going to watch a video, and I chose a sisters video, Amoeba Sisters, if you haven't watched them. I'm sure most of you probably have. While this is playing, I'm also going to get the quickly set up, so you'll see that I'll be writing quickly on the board. And I do recommend watching it. If you, depending on your learning style, I learn by writing and really focusing. But some people are very visual animation type learners. So if you're that type of learner, just sit back, relax, and watch the silly video on DNA replication. I like the Amoeba Sisters because they keep it simple and have fun with it. Oh, I need my iPad actually. All right, hopefully this is going to work.
So much is the ultimate director for cells, and it goes forward. When a molecule has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where? Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, not all cells have a nucleus, such as prokaryotic cells. speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helix, the unzipping enzyme. If you recall that DNA has two strands, you can think of two-stranded DNA. Helix doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds and holds the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start the work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts in a certain part called the organ. Usually this part is identified by certain DNA sequences. At the origin, helix, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, binds the DNA strands to keep them separated. And topoisomerase, you have to slow down when I say that first name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next step. Kinase comes in and makes RNA primers on those strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes to the base thymine, and the base guanine goes to the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3', or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons of the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside this region. Now you do the same thing to the other side, but keep in mind DNA strands are anti-parallel. So let's count these. Again, clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this one. This strand on the left runs 5' to 3'. And the strand here on the right runs 3' to 5'. We'll explain why order matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helix. In this example, it will keep unwinding in this direction. Try
slide on amoeba sisters. Sorry, you're not sober. Make sure you do see the quickly number. That's really big. But hopefully that helps you guys make sense of the 5' to 3' and the leading versus lagging strand, which I know it's just hard to say, but it's easier to just visualize when you see someone animated. All right, any questions? I did say that we were about to talk about, the amoeba sisters were talking about proofreading. So that's what we're talking about next. After we talked about all the DNA replication stuff, mistakes do happen. So mistakes happen, and luckily, our polymerase, DNA polymerase, has a proofreading capability. So it can actually see its own errors if it does make an error, and it has this exonuclease activity. So when it's, for example, right here, it made an error. It put a C with an A, and that's obviously incorrect. And so it can use its exonuclease activity, which allows it to remove the incorrect nucleotide, which was an A, and replace it with the correct one. It can't do this afterwards, but as it's adding the bases, and again, we just use base as another term for nucleotide, so as it's adding the bases, it's actually checking its work. And as it's doing that, if it sees that it put the wrong thing, then it will correct that with that exonuclease activity. And that's DNA polymerase 3 again. So DNA polymerase 3 is that major enzyme that's actually adding the nucleotides, growing the polypeptide, not polypeptide, growing the DNA chain. And again, it's the one that has the proofreading function, which means it has enzyme activity. This exonuclease is an enzyme activity allowing it to remove bases as well as add bases. So, proofreading, exonuclease activity of DNA polymerase. So if an incorrect base has been added, this exonuclease activity allows it to remove it and replace by the correct one. So, and just so you know, there are exonucleases and there's also endonucleases. Endonucleases will cleave inside a chain. Endo means inside. Exo means out. So essentially, it's going to cleave at the outside of the chain. So DNA polymerase's activity is an exonuclease. It can't correct errors inside. Once it's made the DNA chain, it can't go back in and fix anything. And so DNA mutations will still occur. This proofreading activity of DNA polymerase is not perfect. And that is why, as accumulation of mutations occur, your cancer risk will increase as well. Because DNA mutations can cause cells to go rogue, or the cell could die, potentially. So DNA mutations, they can also be beneficial. That's how evolution occurs. As time goes by, mutations occur. And if they benefit the species, then that species will survive and live on, and that mutation will live on. So mutations aren't all bad. But the ones that are unhealthy tend to be eradicated over time with evolution, unless they're not so bad that where the population can still reproduce. So, for example, there are a lot of mutations, genetic issues that still survive, in part because they don't necessarily kill or aren't fatal straight out. Usually the person can survive long enough to procreate, have offspring, and then that mutation will continue, even if it's not exactly beneficial. And this slide, I guess it could have been placed a little higher up, but it's just showing the removal of the RNA primer. Remember DNA polymerase 1, there's 2 DNA polymerases. DNA polymerase 3 here adds new nucleotides. DNA polymerase 1 is what's going to remove the RNA primer and add the correct nucleotides when it's done. So remember, every time you start replicating your DNA, the first thing that has to happen is that RNA primer is put on, and then DNA polymerase can come and start adding nucleotides. But then at the end of that replication, that RNA primer has to be removed, and that's when DNA polymerase will come in, remove it, and add the correct nucleotides. So just so you know, there's two different DNA polymerases. DNA polymerase 1 removes the RNA primer. DNA polymerase 3 is the major one here. This is the one that actually adds the main nucleotide bases to the growing leading or lagging strand, either way. Questions about DNA polymerase 1 or 3? And ligases, what do ligases do? They essentially help fill in any gaps, and they do this by helping form these phosphodiester bonds between the different nucleotides. You don't have to remember that. Just remember that DNA ligase is going to come in, it's going to glue everything together, make sure all the gaps are filled, and all the bonds are correctly formed. And that happens at the end of DNA replication. So in this thing right here, I'm not teaching about this, but remember, whenever you replicate your DNA, it's only because the cell is about to divide. Your cell doesn't just replicate the DNA for fun, it's because the cell is getting ready for cell division. And when you're going to divide into two, you got to replicate your DNA so that each cell is going to have a copy of that DNA. So this is just showing you that during mitosis, that's the actual division of the cell, that's the only time, well, technically you synthesize your DNA in interphase here, and then the cell divides during mitosis. But replication only of the DNA only occurs when the cell is getting ready to divide. But you don't have to worry about G1 and mitosis and G2. This isn't a biology class. Hopefully you're learning that somewhere else. All right, this one, don't memorize. I have this slide for a reason, that you don't have to study this slide. The reason I have this is because DNA polymerase 1 and 3, the one that we talk about in all basic biochemistry or biology classes, is because we understand bacteria DNA replication better, or we can explain it more easily using bacteria as an example. All of these polymerases are actually eukaryotic or human polymerases. And we have a lot more. Humans have a lot more DNA polymerases than bacteria. You don't have to remember these. But I do want you to understand that DNA polymerase 1 and 3 are actually coming from bacteria replication. And we use them because they're simpler for basic biochemistry students. If you were a biochemistry graduate student, you would be having to learn all these guys, but you're not. Just know that there are others for eukaryotic cells. A lot of words in the slides. For telomeres are actually really interesting. We see them in the news sometimes related to health as well as cancer. And so this might be something that you hear about even from the general population walking in. I hear about telomere length. What's that about? So telomeres are at the end of your chromosomes. So for example, here's a chromosome. Well, let me just show you these chromosomes. These are chromosomes. So these chromosomes have at the end are called telomeres. So telomeres at the end of your cells, or at the end of your DNA. And as your cell ages, it's dividing. So over time, as it gets older and it's maturing, it's continuing to divide. Every time it divides, its telomeres shorten. Eventually, the telomeres shorten enough that the cell is dying. And that's one of the, so people are looking at, there's two types of researchers looking at telomeres. And by the way, they shorten every time they divide, essentially because, I have it written out in here, but essentially every time your lagging DNA strand, it's right here, lagging DNA strand is being made, at some point there's no place to put the RNA primer. And so because of the lagging strand, that causes your DNA to shorten every time it's replicated. And that's okay because telomeres are junk DNA. There's no genes coded in your telomeres. I'll say that again. There are no genes coded in your telomeres. They're considered junk DNA. They're just long strands of TTAGGG and repeated over 100 to 1,000 times. And that junk DNA is there because your cells know, evolution knows, that every time your cell replicates, it shortens its telomeres. And so every time, that's okay because your telomeres are just junk DNA. But it also means that as you age, that cell is eventually going to die, which means this human organism will die in part because your telomeres shorten so much. All right, I'm going to take your question first. Because because every time you replicate your DNA, the DNA shortens. Imagine if a gene, a really important gene, is at the end of your DNA. If telomeres weren't there, then that would interfere with that gene. So they are junk DNA, but they're protecting the coded genes that are within that chromosome. Yes. Yes, so that's what I'm saying. Like when there are two types of researchers studying telomeres. One, the type of researcher that wants to live forever. So I used to look at telomeres. I remember thinking, I should have studied telomeres when I got my doctorate. Because we know that if you can maintain your telomeres, and by the way, cancer cells know this. That's the other researcher that's studying telomeres. Because cancer cells have found a way to turn on this enzyme. It's called telomerase enzyme. It will replace telomeres. And cancer cells, the reason they can divide uncontrollably and potentially kill this human organism is because they can turn on telomerase, and that cell can divide infinitely, eternally, and as long as that human organism is alive, it can keep dividing. So some researchers, some cancer researchers, are targeting cancer cells in order to prevent them from turning on telomerase, so that they can kill the cancer cell. And then the longevity researchers are looking at telomeres because we also know that exercise, healthy nutrition, healthy lifestyle will keep your telomeres longer over time than someone who has poor lifestyle habits. So maintaining healthy lifestyle habits have been shown to maintain telomere length longer. So yeah, you have two different researchers. One's trying to kill cancer cells, and they're targeting telomerase and telomeres. And by the way, we all have the enzyme telomerase in our cells, but our cells know we should die eventually. We shouldn't be living forever. And so they don't activate, our cells do not activate telomerase. Only when a cell is behaving abnormally, like a cancer cell, will it activate telomerase. But that is something that can be looked at as far as health and maintaining longevity. If we can find a way to activate that telomerase and do it healthily, then maybe there's some way that we can actually add years to our life from that scientific perspective. But it is an interesting topic. We're not going to talk too much more about it, but that's telomeres. That's what telomeres are, and different types of researchers. Again, because the cell is aging, if we can help prevent that, then we may help prevent aging of the human organism. So cancer cells have a way to turn it on. And then they can just keep dividing until they eventually kill the human organism or whatever organism they're in. And I should just throw out this slide. So this is just showing the action of telomerase. So telomerase is the enzyme that adds back the telomere. And that way the cell can keep dividing indefinitely for however long it wants to continue to divide. And here's the telomerase just adding more telomeres. And some of these pictures, so I want you to understand also what DNA looks like. So remember, DNA is packaged. We have been looking at it as a double helix. So this is my really bad double helix here. But it's long, tiny strands of DNA. And that DNA needs to be packaged properly to keep it safe. And so that's what these green proteins are. These green proteins are called histones. And your DNA, which is in red here, is wrapped around histones. And this is the way that your cell organizes and packages DNA. It packages it around these green balls, which are histone proteins. And the DNA is in red, and it wraps it around. And when you package your DNA around histone proteins, then it's called chromatin. So sometimes you hear DNA being called chromatin. Chromatin is just packaged DNA. And then when you want to super pack it, let's say you're moving, like I just tried to do a couple weeks ago. I say try because I'm still in chaos. But when you move, you have to pack. And so when you're dividing your DNA, you want to pack it even more. You want to organize it even more. So really tight packing and super coiling of that DNA makes chromosomes. So here's the picture. Here's your DNA when it's unwound. Here's where it's becoming chromatin. It's being packed around the histones, those green protein histones. And then you want to super coil it to make chromosomes. And that's what this last guy is. So you actually, even though we tend to visualize DNA as chromosomes oftentimes, chromosomes are not something that your cell maintains all the time. Chromosomes are super packed DNA, and you only make chromosomes when you're about to divide the cell. So again, I just want to make sure you understand the difference between regular DNA, just the double helix, versus being packed DNA. So sometimes if you hear chromatin, it is still DNA, it's just packaged DNA. And then when you hear chromosome, it's still DNA, but it's really packed together, super coiled DNA. So I just want to make sure you understand the differences between those three, between DNA, chromatin, and chromosomes. They're all the same thing, they're just packaged differently. DNA isn't packaged, chromatin is packaged, and chromosomes are super packed. All right, now let's talk about mutations. That's kind of where I was heading before. I forgot there were a few slides before that. So there's two types of repair mechanisms that you need to know. When mutations and errors occur, there are two ways to correct these errors. So there's others as well, but these are the main ones. Mismatch repair proteins. This is the same thing that DNA polymerase has. It's the exonuclease activity. So you have enzymes, these mismatch repair enzymes, that will simply take one nucleotide that's wrong and fix it. But that's only when there's one mismatch. One mismatch, we can just easily go in and fix it using mismatch repair. But let's say we have a bigger problem, like a whole chunk needs to be taken out. And that's what's going on here. You have what's called a thymine dimer. So notice that these two thymines have actually bonded to each other. That's a problem. This is really common with UV rays. UV rays tend to cause these thymine dimers. And that's starting, DNA mutations start occurring, and that's why skin cancer, if you stay out in the sun too long or too often, will increase your risk of skin cancer, in part because UV will create these thymine dimers. And you need a good excision repair enzymes in order to fix this. So even if you are out in the sun a lot, like some people are, then you need what's called these nucleotide excision repair enzymes. Not the mismatch repair, but nucleotide excision repair. And they'll take out this whole chunk and replace it. That's what they do. They don't just replace one nucleotide, they take out a whole chunk and replace it. When you get these thymine dimers that we have, that's what you have to do. So thymine dimers distorts the DNA structure, can cause problems during replication, can lead to mutations, and that's when you need the nucleotide excision repair. So mismatch repair, that's something that DNA polymerase has. You just replace one nucleotide. Nucleotide excision repair, that's where you're taking out like a whole chunk here and replacing it. Thymine dimers are common, but as long as your cells are working properly, they can fix that issue. So again, this is just your textbook Essentially, you have a dimer here, if you create a nick, and then that nick is going to be fixed by the nucleotide excision repair enzymes. This is what happens if you don't have proper nucleotide excision repair enzymes. So if you have a defect in this, so a defect in the nucleotide excision repair, then it increases, then it can cause xeroderma pigmentosa, which is essentially you are not able to repair the damage caused by these dimers. And people with xeroderma pigmentosa have to avoid sunlight. It's really sad. And they have a much higher risk of contracting skin cancer. Essentially because they distort the DNA, which means you're likely to get more and more mutations every time your DNA replicates. And just a couple, there are different ways to get mutations. Some are induced, meaning X-ray, radiation, UV. These are things that are coming from the outside in that can potentially cause mutations. And then there's spontaneous mutations, which can just happen naturally with reactions taking place in the body. For example, your body's all the time making free radicals. Free radicals want to grab electrons from everything, including DNA, and they can start damaging DNA. And these are created naturally by the cell. That's why you need antioxidants to help get rid of the free radicals so they won't damage the DNA. But these are spontaneous mutations. So induced comes from the outside, the environment, whereas spontaneous comes from your own body's chemical reactions. Yeah?
Where does the melanin come into?
Melanin? So there's different types of skin cancer, and melanin is the most dangerous type. Melanoma. So melanoma is essentially, it's the melanocytes that become, they're the ones that are making the melanin in order to protect the skin cells, especially the basal skin cells that are replicating all the time. And so they're meant to protect it, but they become cancerous themselves, and that's why, and the thing is with melanocytes, the ones that are making the melanin, the reason why that skin cancer type is the most dangerous is because that's the one that's most likely to invade other tissues. It's malignant because it travels through the blood. What is that called? Metastasize. It's most easily metastasizes, so it can leave the skin and actually go elsewhere in the body. That's why it's a really dangerous type of skin cancer. Then there's other different types of skin cancer. Some of it's just pretty much easily removed, but melanoma is potentially is the most fatal of them, and it needs to be caught early. If it's not caught early, it's the most dangerous because it can metastasize, which means it can leave the area that it's in and travel to the lung and cause lung cancer, travel to the bone and cause bone cancer. So it travels a lot. That's why it's dangerous. But it's still caused by the same thing. It could still be caused by thymine dimers or anything else. And by the way, there's also silent mutations. Those are mutations that don't actually cause errors in the DNA. Even though it's a mutation, it still codes for the same protein. You don't get errors. Missense and nonsense. And actually, we're going to be talking about these when we get to protein. So you'll see this again. So I'm just going to briefly go over them. Silent mutation means it still codes for the same amino acid, not going to cause a problem with the protein. Because remember, that's what DNA is. DNA codes for protein. And so a silent mutation means the mutation might be there, but the protein's just fine. The same amino acid shows up. Missense or nonsense means that it still not necessarily a problem because it'll still create, well, a missense. Do I have slides on this one? I don't. So let's hold off on these because I actually have pictures. I think this is the last PowerPoint lecture that I'll have where we'll talk about the different types. And there's like 2 other types we need to talk about. But these are going to be more relatable when we talk about protein synthesis. So you'll see these again. Oh, we are done. Yeah. All right, not done. But we might as well go on and take a break now, and then we'll come back and start on RNA. So quickly, we're gonna take 10 minutes here, and then we're gonna start on the next lecture, number 13 on RNA. It should be in week 9.
Oh, yeah.
Oh, so there's this one. So everyone wants to do this action. I was saying. You just say urge. What the fuck is this? I know. But you. Are you saying you right? Are you up there? This is a hard photograph. You can't be doing that shit. You came up. She said she was so that shit's too expensive. Just record it for no reason. That shit like $3, right? I got shirts. I don't know. I got a shirt. You know what? Georgia, that shirt is so. You just be trolling back. You ain't doing shit with your body. This thing was
why do I see this as I would say, out of all the jokes that I've been to, I would recommend you that one. And I even went like 40 minutes late to the time, but they're still like so much. It's very like, I don't know, like everybody in there is just like
And so I told the student, like, I don't necessarily recommend it because they can, like, stuff too fluffy. I don't know stuff that maybe I didn't feel about it. I don't know all the sources. I think it's interesting. And I don't know if I had to ask her to take it. She's a guy who's right? She was trying to step out of her instead of two. She drives a lot. We drive. I had a student tell me that they've been because I don't have like the posture or like that back and forth. I have listened to it really wasn't like if I had a lecture like that, but I had a student tell me because I had these for this class and she said she takes my exams because she doesn't listen to my driving everywhere. So I don't know if this like my videos, and she did that, and that worked for her. Again, she wants to get on an audible. I'm so sorry. Alright guys, we're gonna get started. So again, with this last couple of lectures, it's really hard for me to find lectures on fatty acid synthesis or review videos that are really good. So I don't recommend, they go into way too much detail than you need for a lot of that stuff. So my lecture videos are probably a good resource for you guys when you're studying for the final exam. But when it comes to DNA replication and what we're about to talk about next, amoeba sisters are also really good. So if any of this isn't very familiar and you want it broken down, like really broken down in a fun way, I really recommend them. There's a few good biology lecture series out there. Just can't find really good ones for biochemistry yet. Maybe one of these days I'll have fun making something like that, but not anytime soon. So we are going to be talking about going off of what we discussed, which was DNA. DNA is the blueprint for making protein. So why do we care about your DNA? Your DNA has genes, and those genes are codes for making protein. So genes are just DNA in our chromosomes that are specific, a specific sequence of amino acids which are the building blocks of proteins. So proteins are really important for physiology, for orchestrating nearly every function of the cell. So proteins are really important. How do we make those proteins? We make them via our DNA or the genes, which are going to be the sequence for making those proteins. So this was the central dogma we started with in the last chapter. Central dogma, so this is that we need DNA, which is the blueprint for making RNA. So we first have to transcribe DNA into RNA. So just by the way, these two terms, when we go from DNA to RNA, it's called transcribe. Let's get these words out of the way. DNA to RNA is transcribe. When we are going from RNA to protein, that's going to be translation. You're going to see these terms a lot. Transcribe or transcription. So transcribe versus translate. So again, DNA to RNA is transcription. RNA to protein is translation. And we have to do both of those. We have to do transcription first, though. So we're going to start with talking about transcription, and then we're going to talk about translation. So, central dogma. Essentially, transcription is the cellular process of generating the messenger RNA. So when we take DNA and we transcribe it into messenger RNA, mRNA. So mRNA is the copy of the DNA gene. And then we're going to use that messenger RNA, so that's going to be transcription. So this part here is called transcription. And then the next part is where we take this mRNA and we're going to translate that mRNA into a polypeptide. And polypeptide, ooh, that was loud. I can hear that one. Polypeptides are going to be folded into making protein. I still haven't got my apple pen yet. I've been lazy. So that polypeptide is going to have to be folded into a protein. But when we go from mRNA to polypeptide, what is that called? That is called translation. So those are the steps, and that's my really bad handwriting there. Going from DNA to messenger RNA is transcription. Going from messenger RNA to polypeptide, that's translation. And then we fold that polypeptide and we make a protein. That's the central dogma of DNA protein synthesis. there's more to it than that, so let's get to the next slides. So how do we code for the amino acids starting with mRNA? So a given amino acid, so AA, a given amino acid is encoded by then more than one nucleotide. So let's start with the start codon. All protein is going to start with the same start codon. That start codon that I do want you guys to remember is AUG. AUG. Right here, it's in red. And what does that code for? What does MET stand for? Methionine. Methionine is the start amino acid on your proteins. And the codon that codes for it, so that AUG is the messenger RNA. It's the mRNA code that codes for methionine. You also, so notice here, for example, we're going to look at phenylalanine up here. Phenylalanine is PHE. There are 2 codons that code for phenylalanine. So if you get 3 U's in a row or a UUC, that mRNA sequence codes for phenylalanine. Do you need to memorize that? No. The only one you need to memorize right now is methionine AUG. All of these, serine is a good one here. Serine is coded for by 1, 2, 3, 4 different sequences, 4 different codons or mRNA sequences code for serine. Proline also has 4. Here's 2 more for serine. So notice that you can have different codes that will code for the same amino acid. So serine is coded for by 6 different codons. And a codon is just the mRNA sequence. So let me I'm going to back this up just for a second, because I feel like I'm going to lose people if I don't explain this. All right, DNA. DNA, for example, this is your DNA. You have A, G, C, T, A, T. AA. AG, whatever. So the reason I'm pointing this out, it's in triplets. AAG, CTA, TAG. This is called triplets. So it's called DNA triplets. And then when you copy your DNA to make messenger RNA, it's got to match the DNA. So mRNA, however, is going to be called codons. We don't call them triplets, we call them codons. So when you copy your DNA, what is this A going to be? Is it going to be a T? Because remember, is there thymine in RNA? No, it's a U. So T would have been an answer if we were replicating our DNA. So when we're replicating our DNA, then that would be a T, T, C, G, A, T, etc. But we're not copying our DNA, we're making messenger RNA. So every time you see an A, you're going to have to put a U. And every time you see a T, you're going to put an A. So this is going to be, you guys tell me, U. What is G going to go to? C. So this one? G, A, U, A, U, C. I will ask you at least one question for you to be able to take DNA and make mRNA out of it. Now, we're going to code for the anticodon. So you have something called tRNA, and this is on my slides, and I will get to it. But tRNA are called transfer RNA. So that T is going to stand for transfer, just like that N here stands for message. Transfer or transport. And the reason it's transfer or transport is because it's what's carrying the amino acid. So tRNA carries amino acids. And so it's carrying the amino acid, but it has what's called an anticodon. So it has to match the mRNA codon. And And so that U is going to be matched to a G. All right, what's the next ones? What's this G going to code for? C, U, A, G. So that's how you do. You go from DNA to messenger RNA to tRNA. So you have triplets in the DNA, which code for the codons in the mRNA, which code for the anticodon in the tRNA, which again, each one of these, so AAG here, AAG codes for an amino acid, and that amino acid is going to be carried by the tRNA. So let's see what amino acid this codes for, because I don't have it memorized. So it was AAG. So all we have to do is look at this. Here's so here's A, here is A, and here is G. So if we follow these together, then essentially this is here. AAG, it codes for lysine. Do you have to memorize that? No. But that's how you know what these things code for. That's how you start with the DNA and you can make mRNA. So we'll do one more. Let's see what CUA codes for. So here's C, here's U, and here's CUA. CUA is leucine. So that codes for leucine. So you won't be using this chart. I don't feel like Dylan was trying to put that on the exam, but I do want you to be able to do this. You should be able to do going DNA to mRNA. You should be able to do this translation here, and then going mRNA to tRNA. This is actually the translation part. So does this make sense to everyone? You'll be doing this again. And you'll see this again in my slides. I just wanted to explain it now, since we're talking about the codons that code for specific amino acid sequences. And this is the only one you need to know, AUG, and I want you to know one more. It's a stop codon. So when this codon is reached, it'll stop the mRNA from being translated. And the only one I want you to remember is the one where my undergrad, because it's the only one I remember, UGA. So UGA actually codes for a stop codon. Exactly. And here it is, the stop codon UGA. So it actually doesn't even have an amino acid that it codes for, it just codes for stop. It stops the mRNA sequence. There's two other stop codons. I don't care if you memorize those, but UGA should be easy to remember. That's where your professor went to undergrad. So you need to know two, the start codon and one of the stop codons. Yes. So T, remember thymine is only in DNA. So T's are only in DNA. But it always matches to an A. And A's always match to a T or a U. mRNA RNA uses uracil. DNA uses thymine. So whenever you're going from DNA to mRNA, every time instead of a T, you have to put a U. But if you're replicating your DNA, you stick with T's. So for example, here, if I'm just replicating my DNA, if I'm replicating my DNA, then this A here matches to a T. But if I'm going from DNA to mRNA, then that A here matches to a U because I'm doing RNA instead of DNA. Well, when you're working with DNA, you should know both. You should be able to do it both ways. When you're working with DNA, you always put a T. When you're going from DNA to RNA, you have to put a U instead of a T. So everywhere you would have put a T, you're going to have to put a U, because that's a major difference between DNA and RNA. DNA only has adenine, guanine, cytosine, and thymine. RNA has the three, adenine, guanine, cytosine, but instead of a thymine, it has uracil. So you should know that major difference between DNA and RNA. DNA has a thymine, RNA has a uracil instead of a thymine. And you should be able to do, yeah. I might have a question on DNA replication and you have to match this DNA to a DNA strand. But I might ask the question, translate this DNA into mRNA, and then you're going to have to put a U instead of a T. Does that make sense to everybody?
Sure.
All right. So again, amino acids. You thought you were done. You were not done with amino acids. But we're not going to focus on these guys. You're not going to do any Michaelis-Menten kinetics today. But you do need to see this. The amino acid structure is going to be put together. So here is, remember the structure of protein. We talked about this in Biochem 1. It starts with the linear structure. The linear structure are the amino acids being covalently bonded together. So this is just a linear strand. The secondary structure is, it creates kind of these alpha helices or beta pleated sheets. This is an alpha helix. But it can also make beta-pleated sheets. So this is just folding of the linear RNA, of the linear polypeptide chain. Tertiary structure is more folding, and then a quaternary structure is when you have multiple polypeptide chains. So this linear structure, keep in mind, this is called a polypeptide. If it's not folded, it's called a polypeptide chain. Poly means many. Peptide is just another name for amino acid. Peptide is just another name for amino acid. So this is just a polypeptide chain, and these are the polypeptide chains that are folded. So when it gets to the tertiary and quaternary and secondary structure, that's just the folding of the polypeptide chain. All right, so getting back to RNA and how we're going to copy DNA. These are the types of RNA that you need to know. We already talked about two of them. Messenger RNA is made using DNA as the template. It's used for both transcription and translation. And it uses, RNA uses uracil instead of thymine. Thymine's only for DNA. TNA, and it's called transport or transfer RNA. It's only needed for translation. And it's what transports the amino acid to the growing polypeptide chain. And last but not least, just another definition for you, rRNA. rRNA stands for ribosomal RNA. And it is part of the ribosome. You cannot make protein without ribosomes. So ribosomal RNA is part of ribosomes. Ribosomes are actually, I have this in another slide, but they're protein and RNA come together to make the ribosome. But ribosomes are not completed, they're not active unless an rRNA, the ribosomal RNA, is added to the ribosome. And they do the really important part, the ribosomes do, of reading the codons from the messenger RNA and match it with the anticodons from the tRNA. I can already feel like this is going to be another really good Amoeba Sisters video, this animation, because all of these terms, if you've never seen this before, might be swimming in your head. All right, and this picture is just showing you the ribosomal RNA, and it's got tRNA in here too. So remember, tRNA are going to be carrying the amino acid. And this ribosome here, if you see those blue squigglies, all these blue squigglies in here are the rRNA. They're rRNA. There's different sizes of them. So here's the different sizes of the prokaryotic and eukaryotic RNA. I don't care if you memorize those sizes, but I do want you to know that this ribosome, this thing in green here, cannot be made, this ribosome cannot be made without ribosomal RNA. So you can't make protein without ribosomal RNA. It's going to be necessary for reading the messenger RNA. Here's showing you the transfer RNA. So this is a good picture of the tRNA. It also has a 5' and 3' end. Here's the anticodon portion. So this is the portion that's gonna be carrying the CUA or whatever it might be. And up here is gonna be the site for the amino acid attached. So this is gonna be the site where the serine or the glycine is gonna be attached. Again, tRNA carries the amino acid. That's its function. And you don't have to memorize this structure, but just know that the transfer or transport RNA, tRNA, is what carries the amino acid to the growing polypeptide chain. And it's going to be matching the mRNA codon. Some more terminology before we actually get into transcription and translation. Now you have to know the specific enzymes that are going to be doing the major work. And these you're going to be working on making a few things. So the main one, the really important one, is going to be RNA polymerase II. Yes.
What did you say that it was matching? The codon? Like the tRNA carries the amino acid to grow polypeptide chain that matches?
Yeah. So I'm just going to go back here real quick. So remember the messenger RNA, mRNA, is the codon, and the tRNA has the anti-codon. So it's got to match the mRNA codon. And it's carrying the amino acid. So there are three types of RNA polymerase you got to keep straight. RNA polymerase II is the important one. It's the one that actually is going to be making the messenger RNA. And it's called pre-RNA because the mRNA is going to be edited. So you're going to have to learn about editing a little bit here too. But the messenger RNA is going to be made by RNA polymerase II. The other two, just remember briefly, RNA polymerase I, this one makes your rRNA, which is going to be in the nucleolus. Hopefully you learned about that in cell biology class somewhere. But either way, RNA polymerase I is going to be the enzyme that produces your rRNA for the most part. I'm making it black and white for you guys. And RNA polymerase III is this last one here. It's going to be the one that's making most of the tRNAs. The one that we're going to be focused on most is going to be this RNA polymerase II. It's the one that's going to be transcribing DNA into messenger RNA. So this is the real big one, the really important one. The other two are important as well. You need our ribosomal RNA that's going to be made by RNA polymerase I, and you need tRNAs, and those are going to be made by RNA polymerase III. I'm making it black and white. They do other things as well, but generally speaking, that's a good way to remember them, their main functions. And again, there's different sizes of ribosomes. Don't memorize which is which, but there's different sizes of the ribosomal or rRNA, and it's RNA polymerase I that primarily that's in charge of synthesizing them, making these ribosomal RNAs. RNA polymerase III, again, that's the one important. It doesn't make the messenger RNA, but it's the one that makes the transfer or transport RNA. So this is also important for making the transport RNA, and which is going to be carrying the different amino acids, whatever they are, like glycine or serine or methionine. But this is the one we're going to focus on for the most part, because this is the one that's going to be making the messenger RNA, which is going to code for the protein that you're going to make. Your cell is going to be making. All right, we're going to start transcription. So first thing you need, so this thing here in blue is your DNA, and the red here is going to be your messenger RNA. So you see that you're creating this bubble here, this transcription bubble, so that you can get the machinery in here in order to start making your messenger RNA strand from the DNA. So it's going to be an exact copy of the DNA, except instead of thymines, you're going to be using uracil. So there's 3 steps: initiation, elongation, and then termination. And again, we're focused on transcription. There's 2 parts of protein synthesis: transcription And the other part's translation. So we're gonna focus on transcription first. I know you guys, some of you don't, especially don't have a lot of background in this, so I'm always trying to figure out the best way to describe this. But when it comes to messenger RNA, making messenger RNA, you have to look at the gene. So the gene is the DNA that codes for a specific protein. For example, this could be the gene for insulin. So let's say insulin is just a protein hormone. Let's say it's the gene for insulin. In green here is the actual coding sequence. The coding sequence, this +1 right here, is going to be the beginning of that gene. But there is an important part upstream from that gene. And that is called the promoter region. Promoters are upstream of the transcription start site. And these promoters are going to be really important in regulating transcription of that gene. All right. So the initiation site is going to be where you start with that transcription. Upstream of that is going to be in a negative number. And this is where you're going to have the promoter region. So the promoter region is going to be a negative number up here. It's going to be a negative number. And then downstream is always positive. Downstream of the gene are the nucleotides following the initiation site, and they are always going to be a positive numbering. The reason this is important is because if you go into any site and look for a gene, it's going to give you what number it is in that chromosome, or what number it is in that gene. If it says a plus number, then you know that we're talking about inside the gene. But if you're reading that paper or whatever it is you're reading, and it gives you a negative DNA number, it's talking about a promoter region that's upstream of that gene, but regulates that gene. So promoter regions regulate the gene. So they are important even though they're a negative number and they're upstream of the gene. So we'll see this as we look at the slides. All right, so again, we're going to have this transcription bubble. Here's the bubble here. And that bubble is where you're going to have RNA polymerase coming in, and it's going to start transcribing this new, this mRNA that's going to be an exact copy of the DNA, except that you've got uracils instead of thymine. You have a uracil instead of thymine. RNA polymerase, however, is going to attach to the promoter region, and that makes sense. It's not going to it's not going to attach to the beginning of the gene. You might miss the first part of the gene. So RNA polymerase is going to land on the promoter region. And the promoter region, like I said, is always a negative number. So this negative 10 means it is negative 10 nucleotides upstream from that +1 start site. This negative 35 region means that it's negative 35 nucleotides upstream from that +1 start site. And that's where the RNA polymerase is going to land. It's going to land on the promoter region in order to start transcription at the beginning here. So RNA polymerase attaches to the promoter. Specifically, it attaches to what we call the ta-ta box. The ta-ta box is right here. I'll put it in green. This is the ta-ta box. And that's where, that's a major place where the promoter region is gonna, where the polymerase is gonna come land on the promoter region. Yeah.
So you said with the negative numbers,
Mhm.
right, so let's using like negative 10. You
Yep.
said it's negative 10 like codons from +1.
Nucleotides.
So just like bases and stuff, so T, E1, A2.
Yep, exactly. Exactly it. And that's how scientists, medical professionals can talk to each other about genes. You can say site +200, and you know where that person is talking about. Or you can say site -200, and you know that they're talking about upstream of that site. So it's just a way for scientists and medical professionals it's the part of the scientific language of how we communicate about genes. And it's just kind of this common knowledge that you guys will, if you ever have to do this, you'll remember it. When you have to actually go look at a paper because you have a client who has some defective gene or something. Maybe that'll happen. All right, so again, the DNA sequence in which the transcription machinery binds is going to be on that promoter region. So transcription machinery is going to bind to this promoter region. Promoters are very important. They decide whether the gene is transcribed all the time, some of the time, or not at all. So promoter regions are the regions that regulate whether or not that gene is going to be coded for or not. And we'll talk about regulation later. Well, actually a little bit now. So here's talking about more of the promoter region. Again, the ta-ta box it's the reason it's called a ta-ta box is because it's a lot of T's and A's, ta-ta box. It is located usually around -10 to -25 or 35 upstream from the initiation start site. It's going to serve as the binding site for transcription machinery. And that transcription machinery is going to include transcription factors. Transcription factors are the factors that help regulate gene transcription. And so here you have the promoter region, you have the ta-ta box, and you have these TFs. TF stands for transcription factor. And these are TF2. TF2 stands for, this 2 part stands for RNA polymerase 2. I actually have this in the next slide. Here we go. Transcription factors. Transcription factors, we just tend to abbreviate them as TF. And transcription factors, TF2s, are specific for RNA polymerase 2. So remember, RNA polymerase 2 is what's making the messenger RNA. And so RNA polymerase 2, there's a lot of transcription factors, hundreds, maybe thousands, I'm not sure. But they all have specific functions. The ones that are TF2s are important for recruiting, that's the word I was looking for, recruiting the RNA polymerase 2. In other words, here's the promoter region, and here's the ta-ta box. These TFs, so here's TF2, here's another TF2, TF2A, TF2D, TF2B. These are all have to bind first, and then once they bind to the promoter region, now guess what? RNA polymerase 2 can bind. So without these TF2s, these transcription factor 2s, RNA polymerase 2 couldn't bind. And so you need these transcription factors to help regulate gene expression, to regulate whether or not that RNA polymerase can bind. It can't bind unless these transcription factors have bound to the promoter first. So here you go. Once they've bound, now you can initiate transcription. Once all these, there's lots of them, don't remember which, and they all do different functions. Don't have to know their other functions, but they're all going to help recruit RNA polymerase 2. And then once RNA polymerase 2 is there, you can start transcription and you can start making that messenger RNA. That is what RNA polymerase 2 is doing. It's making the messenger RNA. So there's another region that's upstream, well, it's not just upstream, it can be downstream, it can be in the gene itself. These regions are called enhancer regions, and there's a whole area of science just on enhancer and promoter regions. I don't want you to get bogged into any details, but I do want you just to understand that enhancer regions, first of all, can be anywhere. They can be upstream, they can be downstream of promoter regions, but they do exactly what it sounds like. They enhance RNA transcription from wherever they're at. Do you have a picture? Here's one. So here's an enhancer, and it was upstream of the promoter region, and basically this folded onto it. And that's how it is enhancing transcription. So it's exactly what it sounds like. That's all you have to remember about enhancers, is that they'll enhance RNA transcription. And they can do that no matter where they are in the chromosome, in the DNA, they'll find a way to fold up and stimulate. That's what it's doing here. It's stimulating these transcription factors at the promoter region. So enhancers can be found far from the gene, upstream, downstream, lots of places. And this is what I want you to know. Their function is to increase or enhance transcription of the gene by providing sites for activation of these transcription factors. They just enhance transcription, period. That's really the gist. So don't get bogged down in the details of these. And here's the mechanism if you want to read more about it, that it just essentially loops into the DNA and helps stimulate these transcription factors. So here's just showing that enhancer regions can be anywhere in the gene. They can be upstream, downstream, they can even be inside of the gene. They can be thousands of base pairs away from the gene. No video for you. You already had your video today. We will watch a video next week or tomorrow when I put more of this together for you guys. I want to put it all together and then we'll watch something.
Woo!
So here again, you have the transcription bubble. The blue here is the elongating mRNA strand. And by the way, it happens in the 5' to 3' direction too. So just remember, everything is happening in the 5' to 3' direction. I don't think anything happens in the other direction. It's all in the 5' to 3'. And so here's the 5'. You're making 5' to 3' direction. 5' end of the messenger RNA. Here's the 3' end. And here's the RNA polymerase. So again, here's the DNA and here's the RNA. And it has to match the DNA perfectly or you get a mutation. So this A, again, we're dealing with RNA, so it always matches to a U. G's always matches to a C, and T's will always make an A in the RNA. So again, it's the same. You have 3 hydrogen bonds between the G's and C's, and 2 hydrogen bonds between the A and U's, or the T's and A's. But that is how you can actually form these base pairs while you're making that RNA. So it's going to be an exact copy of the DNA, except the fact that there's going to be uracils instead of thymines. There's also going to be a few regulations that we didn't see with DNA replication. One of these is 5' capping with this 7-methylguanosine cap. So here is the 7-methylguanosine cap. You don't have to remember its structure. That this 5' cap is going to be important for the nascent mRNA to prevent it from being degraded. So the 5' cap, that 7-methylguanosine, is important to prevent degradation of the mRNA transcript. So prevents degradation of the mRNA transcript. And I just want to write mRNA transcript because that's what we're making here. We're going to be making an mRNA transcript. That's what RNA polymerase is doing. And the other thing that this 5' cap is important for, not only does it protect the new mRNA, but it will also help the ribosome identify it. So this 5' cap is going to help the ribosomes identify it for translation, which will be happening later. So we have a 5' cap for its protection, and here's the rest of the messenger RNA. And then at the end, we're going to have a poly A tail. So the 5' cap and the poly A tail, both of these are protecting the mRNA transcript. The poly A tail is also going to help keep it safe. So just so you know, there's a 5' cap at the front end, the 5' end, and at the 3' end, the end of the mRNA transcript is the poly A tail. And both of them are protecting that mRNA transcript. All right, you'll see that 5' cap in poly A tail later when we talk about regulation. But we've started initiation. So how did we start initiation? Essentially, we had the promoter region was bound by the transcription factors, which then recruited the RNA polymerase II, and now that was initiation. Elongation is where you have the assembly of, after formation of the initiation complex, the polymerase is going to be released from the transcription factors, allowing elongation to proceed. So the transcription factors were just there to recruit the RNA polymerase. Once the RNA polymerase is in place, the transcription factors go away and the RNA polymerase can start doing its job. So, and what is it doing? It's making the mRNA transcript. So RNA polymerase II transcribes the major share of eukaryotic genes. So we are talking about eukaryotes. We're not talking about prokaryotic bacteria anymore. So RNA polymerase II is the major enzyme that's going to make the messenger RNA. And elongation is really that simple. We're just elongating it at this point. Eukaryotic cells are a little more complex, so but as far as details go, that's as far as we need to go. But keep in mind, we do have to unwind the DNA. Because remember, the DNA is packed around these histones. That's why we make chromatin. If you have packed up your DNA, you can't express it. And so you have to loosen up the DNA. And so you have to, these histone proteins, you have to unwind the DNA from them in order to express those genes, in order to get the transcription machinery in there to make copies of that DNA. You've got to loosen it from those histones. And so we do have HATs and HDACs. All right, so HATs, histone acetylases. Where do I see? I don't have that. Oh, histone acetyltransferases, and HDACs are histone deacetylases. I'm going to go back to that picture again. So HATs and HDACs. Oh, here they go. Histone acetylases, acetyltransferases, sorry, keep doing that. And HDACs are called histone deacetylases. This is really important for epigenetics, and we'll talk about it a little bit more next week. The HATs and HDACs are going to either prevent genes from being expressed or they're going to allow them to be expressed. And that's by acetylating the histones. So remember the histones are these, I call them balls, but they're proteins. These histones are proteins. And when they're acetylated, then they're going to allow that gene to be, it's going to relax the histones. So acetylating histones relaxes them, allowing for expression to occur. One of my advisors in graduate school studied HDACs because you can actually turn off genes by acetylating the histones. And some disease states can be caused by acetylation or deacetylation. So when you deacetylate the histone, in other words, you take off the acetyl group, it tightens the chromatin, and when you do that, you can't express that gene. So here's an acetyl group that's been added to the histone. And that acetyl group is going to loosen the chromatin, allowing you to express that gene. So this is it in a snapshot, the difference between HATs and HDACs. When you acetylate the histones, you allow expression. That's the gist. When you deacetylate histones, then you prevent expression because this is going to condense the chromatin. Condense is just a sciencey, fancy word for meaning when you pack it together. So condense means you pack something in. Decondense is when you loosen something. And so when you relaxes the chromatin. You could also just say that you de-condense. You might see condense on my exam, so know what condense means. Condense means you pack something together. Decondense means you relax it. And that's just, again, typically used when we're talking about, well, a variety of biology things, but especially related to DNA. So relaxing chromatin, you decondense it. That's what acetyl groups do. You throw an acetyl group on there, you relax the chromatin. You take off the acetyl group, you're going to repress gene expression because you're going to pack together or condense the chromatin. So HDACs and HATs play a major role in epigenetics, which hopefully we'll have time in a little bit to talk about next week. But for now, at least know what HATs and HDACs are. Oh, so gene activation versus gene silencing. That's the effect that you need to know. So HDACs will silence the gene, keeping it from being expressed, whereas acetylation will activate the gene, allowing it to be expressed. So what your grandma ate and your great-grandmother and grandfather ate actually play a role in whether or not your genes are acetylated or not. So it plays a role in who you are today. Again, epigenetics. Hopefully we'll have time to talk about it next week. All right, so we've elongated the mRNA transcript. See, it's a lot simpler than DNA replication. So we started with initiation, which is where we had the RNA polymerase come in with the transcription factors. Elongation, where RNA polymerase was released from the transcription factors so it could elongate and make the mRNA chain. And now termination, there's a couple of different ways to terminate it. But we're just going to talk about one of them. Row-independent termination. Row is a protein that can be used in bacteria to terminate transcription, but eukaryotic cells do not have row-dependent termination. So we just call it row-independent because it's not using a row protein, just in case you're wondering where we got the word row before. Row comes from bacteria. But eukaryotic cells use a row-independent termination, and that comes from a CG nucleotides that bind together to make a hairpin. And that's what this picture is showing you right here. Here is the RNA transcript that is being made. And essentially, as it's being made, there's this GC-rich loop, or GC-rich area, that's going to fold back on itself and make a hairpin. And that causes RNA polymerase to fall off. So it's kind of that simple. It's almost too simple, it sounds hard. Row-independent termination just is caused by an area with lots of G's and C's. Those G's and C's fold back on themselves, making a hairpin, and that causes RNA polymerase to fall off. And that's one of the ways that termination occurs. And I think it's the only way that I'm making you guys learn. Yep, we're only going to talk about row-independent termination. It's the primary way. And that's going to stop transcription. So row-independent termination, that is going to stop transcription. And you can see the G's and C's here. There's a lot of G's and C's, they fold back on themselves, and that causes RNA polymerase to fall off. Row-independent termination. And so it says prokaryotic here, but it's also a very common way in eukaryotic cells too. All right, so we terminated it. We got the 5' cap, we got the mRNA, and the poly-A tail. So what makes the poly-A tail? It's poly-A polymerase. Poly-A polymerase is going to tag this mRNA transcript with a poly-A tail. And the poly-A tail is called a poly-A tail because it's mostly made up of A's. All right, so we've officially created the pre-mRNA. This is pre-mRNA. The reason it's not mRNA yet is because it has to be edited. And that's where introns and exons come in. So pre-mRNA has exons and what's called introns. Okay, it's not in this slide, but it's probably in the next one. Here we go. RNA processing. It exons, I used to didn't like, I thought it was not intuitive, but exons, oh, here it is. Exons, X means expressed. These guys have to stick around. The non-coding areas are called introns. So look at this RNA transcript. This is the pre-RNA. It's got these green areas here called introns. And they have to be spliced out. We literally call it that. You splice out the introns. What splices them out? It's going to be called a splice zone. Spliceosomes splice out introns. And that then will make the official mRNA transcript once you get rid of the introns. So I think I have that in the next slide. So here's a picture of RNA processing. You essentially splice out the introns, and then the spliceosome is what's taking out those introns, and it actually will put the exons back together. So when you have these exons here, once you get rid of the introns, those exons, so here's exon 1, exon 2, and exon 3 are brought together. So this mRNA is now complete once you remove the introns. So now we have mRNA, it's got the 5' 7-methylguanosine cap. You got the mRNA with full of just exons, and then you have the poly A tail. And that's going to be the mRNA transcript that you're going to be using for translation. This is just showing you more on splicing. So remember, splicing is how you get rid of the introns. So here's the intron. It needs to be spliced out, and that's going to be using a spliceosome. Spliceosomes are just enzymes, well, proteins and RNA, that they're going to splice out the introns. So here's just another picture of that. So a lot of my slides are just, I'm showing you the picture again. Here's the exons. Oh, wait. Oh, by the way, you can actually make different transcripts based on splicing. So some of the variability in me and my sisters and you and your siblings and you and everyone else is the way that your DNA or your RNA is spliced. So my genes, my proteins, are going to be slightly different from some of yours, not because our DNA is that different, because it actually isn't. Our DNA is very similar to monkeys. So what are some of the things that can make us different? Part of the variability comes from splicing. So we have different splicing. I have different ways that my proteins are being spliced, so that my proteins might be slightly vary from yours, even if our DNA is very similar. So some of the variability that we get in humans and different species comes from how we splice various proteins. And that's what this picture is showing you. So here's showing you these different exons, and they're spliced differently. And then here's these exons, and it's the same protein, or it could be a different protein that's just spliced differently. So it started with the same transcript, but these two have been spliced differently. And that could, that's one of the reasons we have variability in this protein functions really well, and this population of people don't have an enzyme that works as well, or maybe it's beneficial in different areas, but it was just some of that variability comes from splicing. Splicing, if you want to watch a video on it. But we're not, we are going to go home. I'm still going to make review slides for this one, but the other two that I've already done